Thank you for that introduction, and thank you to the organizers for inviting me to speak today. So I'm going to talk about the effective and efficient utilization of gene bank resources, and I'm going to focus today on maize because it's a crop where we have got a lot of advanced work over a whole century of advanced work in maize. And so if we're utilizing the gene bank, there have been lots of attempts to do this already, lots of concerted efforts to use gene bank resources. And now we're at a stage where we have an elite population that's very, very distinct from the land races. And also, we've probably found most of the large-scale variation that will be useful to us. And now we're going to be looking at, essentially, we're looking at quantitative traits, which sets us a different, which means we need to maybe take a much more quantitative genetics approach to optimizing our use of these resources, more than just to sort of go into the gene bank, find something good, and make some crosses, which would bring you a benefit 50 years ago. But nowadays, we've got stronger challenges. And I want to talk a bit about how we can be using quantitative genetics to overcome those challenges. Okay. So just a brief introduction. I know many people here know all about CIMMYT, and there's lots of my colleagues here. So CIMMYT is the International Maize and Wheat Improvement Center. We're a non-profit international applied agricultural science organization. And we're basically work on providing improved agronomy and improved production systems, and also improved basic cereals such as maize, wheat, triticale, sorghum, millets, and so on, particularly for the global south. So basically, CIMMYT lines are, CIMMYT germplasm is the dominant germplasm across much of the global south, and even, for example, in the global north, so in the United States, about 60% of the wheat crop grown there has CIMMYT germplasm in it. So we've really had a large impact over the years with our maize and wheat programs. But the main aim of CIMMYT is to enhance the livelihood trajectories and resilience of resource-poor farmers. Whenever we have a meeting, our Director General is always farmers, 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 and that's where we're looking. Is we want to improve the livelihood of farmers, resource-poor farmers, in a way that's productive but also doesn't damage the environment. Okay. So as I said before, the CGIR, of which CIMMYT is a part, so we work on maize, wheat, and dryland crops in Africa, but there are other institutes that are part of the same organization that work on rice, cassava, root crops. All the major crops are worked on by CGIR as an institution, and 80% of the germplasm exchanged by the public sector globally is within CGIR. And within that, 40% of all germplasm exchanged globally per year comes from CIMMYT. And this is coming from our maize, mostly coming from our maize and wheat breeding programs. Okay. And this is just an example of where we send seed each year. So I'm going to focus on maize today, but the things I'm talking about apply to any advanced crop. I could have used wheat, but there's better examples in maize for what I want to illustrate today. So I'm going to focus on heat and drought effects, because a lot of our work nowadays at CIMMYT is focused on adaptation to climate change. So as you can see here in this diagram, drought, this is an example of this is trials under drought management and what a increase in global temperature means in terms of yield change for 1 degree C of warming. So in an optimal environment, optimal management, it goes fairly steady up to about 22 degrees and then starts to decline. This is for maize. But in the drought conditions, the drought is interacting with the heat, and this is much more of a linear decrease. So we need to deal with both of heat and drought effects, and this is becoming very urgent. So, for example, the El Nino drought in 23 and 24 was a disaster in southern Africa. It's their worst drought in 100 years and massive crop failure. And at CIMMYT, we're supplying the germplasm resources for most of these areas, and we need to be able to adapt very fast to this. So what I'm going to talk about is what we've done in the past to utilize gene banks, gene bank resources to improve our material, but then specifically what we need to do now to go faster. Okay, so we have the largest global maize and wheat germplasm bank, so 140,000 wheat accessions, 29,000 maize accessions. So we do the breeding and we have access to all of the germplasm material we need. Basically, this covers essentially everything we need is in our gene bank. So how do we make this 10,000 years of evolution work for us? Is the question. So initially, up until about the late 1980s, CIMMYT was running recurrent selection programs with breeding populations and then occasionally using germplasm material to make synthetics, to make crosses, and looking basically, it was an ad hoc process. It was effective, but it was ad hoc. And so the idea came about 15 years ago of starting a more systematic strategy of exploiting the germplasm bank. And I'm going to talk about two components today, the discovery and the deployment. So in the discovery, we want to what do we use to discover alleles that are useful for drought, heat, or tolerance, or other traits? Do we look at landrace performance per se? Do we make test crosses? How do we use, for example, GWAS to find new allelic sources? And so we are talking about discovery there, and then deployment. What is the best strategy for introgressing this material into elite germplasm? So I'm going to work through the historical look at discovery first. So when we first started to standardize and make a systematic approach, it was a project called Seeds of Discovery, or SEED, and this was largely funded by the Mexican government with a few other partners. And this was a very large-scale project to exploit the germplasm bank data. So the first step was to go into the gene banks and validate the passport data for all of these. So use GIS techniques, do GIS approaches, and painstakingly work through the landrace accessions, see if the information provides us is consistent on the passport data, plot those, understand where they came from. And then when we have that information on reliable passport data for the accessions in the gene bank, we can collate all of the climate and soil variables from those collection sites. And when I'm talking about climate at those collection sites, I'm talking about the climate when that happened, not the climate today. So we can collect temperature, precipitation, relative humidity, vapor pressure deficit, aridity index, and so on. And the idea is we collect variables. This is an informed process. So we take the knowledge of what's important. So, for example, vapor pressure deficit around anthesis is important in maize, so we will focus particularly on those key traits that have been identified by crop growth models or just general knowledge. So first pass, we collect the GIS information, then we have information on the race, the altitude, the climatic information, et cetera, breeder knowledge as well. And we can select a set of land races which appear to be growing in drought environments that seem to have some resilience to drought. So that's one first step, and then we can start using this material to make crosses, and I'll explain how we use that in the deployment in a bit later. But basically, this is the starting point. Get some lines that we know seem to be adapted to drought from the climatic information we have from the locations they came from, and that's one starting point. A second starting point is we can go from the genetics angle. So we genetically characterize the whole germplasm bank. So for maize, that was basically 25,000 land races, a couple of thousand varieties and composites and things from our breeding pools, some wild relatives. CML is a CIMMYT maize line, and also material from elsewhere in the world like PVP crops, PVP lines, and so on. And we count, so in total about 35,000 entries. So in the initial phase of the project, we genotyped all of this with GBS. There was about 1 million markers, which filtered down to around 300,000. Subsequently, we've used DartSeq, both SNPs and silico darts, so the presence-absence variation. And for the GBS data, we collected single samples from land races, and you'll see why in the subsequent slides. For the DART-seq, which we've done more recently, we made composite samples. So we're really sampling a landrace properly by sampling 30 random individuals and getting SNP allele frequencies from within landraces. So we can see if a landrace is heterozygous or a homozygous at every SNP. Excuse the cough. And then from this, one initial approach in this project was we'll take core subsets. So we collect, we understand that here we have the overall variation. This is a three-dimensional figure. The details aren't that important, but this is all the lines that were in the CIMMYT maize gene bank, so 15,000 landraces. And we can make a subset of those that are from strongly drought-prone areas and make a core that represents the drought-prone lines that are within this, so make a core set. And then we can also break that up into the different adaptation zones, so we can make a subtropical core, a tropical core, a highland tropical core. So we can make different core sets for a total of 4,000 lines for different uses, so these sample, appropriately sample the overall variation. And like I said, different adaptive sets, so different drought types, different heat types. So for example here, when you look at the drought, the clusters of drought-resilient lines, genetically they cluster into 6 different groups. When you separate these groups out and take a look, they actually have slightly different kinds of drought. There's drought in humid areas, drought in dry areas, there's strong drought, medium drought. The genetics clusters with the adaptations, clusters with the phenotype, suggesting we are looking at adaptive traits here, which is good because we want to be able to breed for this. And we can also make separate clusters based on this information. So then we want to evaluate this material. So here, this is what I said, this is the GBS data. We have one individual per population, genotyped, imputed, and we have the individuals per se, and then we also can cross to generate F1s, and then we can grow those F1s out. And we will, when we're doing this crossing, if we have a line that's from a high-altitude environment, we'll cross it with a high-altitude elite accession to test in the right environmental and genotypic background. Okay. So we can do the phenotypic evaluation, and we have 2 options here. So we have 4,000 accessions. We can cross to a tester and do the evaluation on the F1, or we can do the direct per se evaluation. And this is what was done in the project. So we have, in the end, we planted 35 trials, 14 locations, 1.5 million data points, because this was the 4,500 core lines, and measured for multiple traits. So this is basically an enormous phenotyping experiment. And this is where the first step, where quantitative genetics comes in, and I'm going to talk about this in relatively short order because our hosts here were the people who wrote the paper, so Gregor. So the first question is, what is the best, if we're doing this discovery phase, what is the best way to carry out this discovery so that we create material for pre-breeding that is most useful? So the first question might be, is it better to evaluate on the F1s from the test crosses or directly on the landraces? And so this is Gregor's work. I'm going to not go into too much details throughout all of this early strategy work. I'm not going to go into too much detail. So what's the best approach to initiate a pre-breeding program? Do we use selected landraces, maybe doubled haploids of the selected landraces, or test cross of elite hybrid and selected landraces? And we can vary some of the genetic parameters of the landraces and phenotypes. So we can vary the genomic variance, so it's 1,000, is the effective population size 1,000 or 100,000? What's the variance in the landrace, different heritabilities? And some of the potential logistic factors like the genotyping platform, the number of seeds you use from a particular landrace for training, the selection of how many landraces you select in the first place, and so forth. And what we're trying to do here is, what Gregor was trying to do here was use genomic prediction to basically evaluate the quality of subsequent bridging germplasm. So that basically and this would be like the mean of the true breeding values after selection, the accuracy of the evaluation, and whether it's similar to the tester. Because we don't want to be selecting elite material, if we use hybrid times selected land races, we actually want to select on the land race material, not recreate the elite. And so the conclusions of this work was that accuracy of genomic predictions are useful. So a genomic prediction approach is effective for finding the most promising germplasm bank accessions for use in pre-breeding. So we can gather some phenotypic data on this core subset and use that to predict the most promising lines in the gene bank for future exploitation. The pre-breeding program should be initiated directly from land races, so should take the data from the land races, not the crosses to the elites. Because otherwise, if you initiate from test crosses, you can rapidly reconstruct the elite donor genome. And then for the other parameters that we discussed, the logistics and the heritability and so on, they created decision trees like the one here to enable the best decision making, to make good decisions on how to on the best discovery strategy. So we can also say, well, that will get you some good lines to start making crosses with and to start working with. We can look directly at the loci using GWAS, and this was successful. We can, for example, here there's an inversion that affects flowering time. You can have a look at the way that the haplotype here is clearly different from these two, and you can plot this in different ways for day strand thesis. And at the time, this was the largest effect on flowering time documented to date. And this was a gene that's in the gene bank that hadn't been exploited before. And you can do this for many traits. So here's you can get successful GWAS hits for grain protein content, for example. And this successfully generated some QTLs that we can potentially introgress into elite breeding populations. But in this project, we also started environmental GWAS. So environmental GWAS is where the environmental variables, temperature, precipitation, etc., are used as traits in a GWAS analysis, as many of you know. And this can be general, so vapor pressure deficit during the growing season, but we also wanted to be more specific, informed by crop growth models and biological knowledge. So we want to look specifically VPD in the month of flowering. And we need to take account when we do this of management, so irrigation, fertilizer, fungicide, etc., that might have been used in that area. So this is less of a problem in maize, but in wheat, for example, if we took a naive approach and we looked at the wheat germplasm bank, we find areas where there's basically no rainfall, but wheat's still grown there. There might be drought adaptation, but those are irrigated locations, so we need to take account of that when we're making these analyses. And this also works well. We can find a few nice large effect loci that are related and relate them to genes that are in that region that have a clear relevance, like a heat shock protein in a heat case. And take that a bit further, we can find these loci, we can fine map them a bit, we can narrow it down to gene, we can look at that gene particularly in drought trials, we can look at expression data, characterize it, and we've got a set of QTL that are useful. But these large effect QTL are rare. There's really not that much. The gene bank has been exploited sufficiently that there's not a huge number of these traits that are available for use, and most of the variation is quantitative, so how can we use that? But we did show that, combining GWAS data with genomic prediction, we can select the most promising lines for the creation of pre-breeding populations. Okay. So moving on to deployment in this initial project. This is the deployment model we've been using at CIMMYT for the last 15 or so years. So we sample about 75 individuals from each land race, characterize which heterotic group they belong to, and then cross to the opposite heterotic group, test and select some F1s, make a backcross, usually backcross, just a single backcross, and we do all of this early development specifically in the stress environment. So if we're bringing in individuals we think are good for heat, we will do these early pre-breeding population development in a hot environment. And this filters out a lot of material because, as those of you who work on maize know, crossing land races to elite material, you get a lot of junk and a lot of failure. And we clean that up, and we clean up a bit on the stress as well at the same time. And essentially, we do a BC1 and then several selfing generations, and we do test crosses at most at BC0, S2, S3, and test these in stress, and we test in optimal environment to see if there's any G by E or effects there. And at the end of this process, we've whittled down, maybe we're doing 50 lines a year, we've whittled this down to 1 or 2 lines that come out at the end. They've been tested, they have yields that match the elite, and they also have some advantage in the particular stress environment, so they might provide a higher yield on average in heat environments. And we call these CGMRL lines, and for the 2 different heterodic pools. And this is material that's pre-breeding material ready to go. So if you, and we send this around the world, so if you're a breeder in Africa who needs some heat-resilient material in an elite background, you can get sent this material and use that in your breeding program. Just a note on the testing, we do this across Mexico's got a fantastic range of environments from different altitudes, different heat, drought, and so forth, so from very humid, hot places to dry, cool places. So we have a range of environments that we can use. We can develop this material quite effectively in Mexico. We do test in other countries later, but this is all done in different sites in Mexico that are good for different kinds of heat, dry heat, humid heat, for example. These are the drought sites. And these are the nurseries where we develop the material where we're testing the stresses as well. And this works. So, here's got a BC1F3 from the subtropical population, run against 3 testers. The red is drought-tolerant commercial hybrids, and then these are, you can see there are lines that are yielding better under drought stress and still yield well under irrigation that are out here that are excellent for use that have come out of this program at the BC1F3 stage, so we've been successful. And this program has been ongoing. Each year more lines were added, and it's a cyclical process that takes about 5 or 6 years. And we create material, we send it to people, it's got drought tolerance. So what we wanted to do was take some of these approaches a bit further. So the phase 2 is a project, an allele mining project. This is a much bigger project, so we are moving this approach to 6 crops, not just maize, 6 crops across the whole of the CG. Maize, cowpea, sorghum, beans, rice. Okay. And now we're going to do environmental GWAS on the whole gene bank, all 17,500 land races rather than just the subset. We're going to use the DART-seq data, so we've got a couple of 100,000 SNPs, 250,000 presence-absence variants, and we are working with partners to impute up to a practical haplotype graph level where we can get over 100 million markers potentially in this approach. The strategy we're using is more a quantitative strategy here. So for each environmental variable, this is discovery, sorry. We collate the top 1,000 hits from landrace GWAS. We calculate the frequencies of those positive alleles in the elite germplasm. If the allele's already in the elite germplasm at anything except a trivial frequency, we're not interested in this project per se. We're focused on material that's in the gene bank that we haven't exploited in previous projects and that's not already an elite material. That turns out to be about 7 or 8% of the total alleles we detect with GWAS have not been previously utilized in elite germplasm. The next step, find the set of land races, the smallest set of land races we can that contain all these positive alleles, because we have everything genotyped, and we focus on having land races containing a high number of positive alleles. So what we're trying to do is concentrate these alleles that we think are positive in the smallest number of land races and use these for future deployment. And so far, in 3 rounds of this analysis for several traits, we've collected about 321 land races containing over 400 potentially useful alleles. And we can validate some of this approach, so we can predict from the environments where the lines were collected. We've got genotype data, we can make a prediction, and we can see that if we make a prediction based on the genotypes of all the lines that were collected from different environments of different temperature, elevation, precipitation, and so on. And then we grow these we grow a bunch of other material in a separate trial. We see that we can predict quite well how well they will do in different environments. And just some examples here, this is a drought example, and these are elevation, temperature, and another precipitation example. We can usually, this is a kind of a good validation in that we can predict future trials based on the information we used in the environmental GWAS. What we can also do is all the loci we found that are significant but already in elite material, when we've grown that elite material in all these trials in the Seeds of Discovery project, we can go back to the historical trial data and say, did these loci we find contribute to drought resilience in those trials? And we're in the process of doing that. Basically, this validates because there are lots of adaptive traits, adaptive alleles in there. So, also with partners in the project, I'm going to go through this fairly fast actually, we can see, is this environmental GWAS the best approach, or is it better to find the best adapted germplasm, do QTL mapping populations with that, and then identify beneficial alleles in a two-step process or one-step? I'm going to cut through that a bit, but basically, the more polygenic the architecture of your trait, there's a tendency for the crossing into QTL, crossing and doing QTL analysis and then using that information is more effective more times than it's like a draw with the environmental GWAS. So we are exploring ways in which we can also use methods other than environmental GWAS. There are some other challenges with environmental GWAS. We've been using quantitative genetic approaches to quantify here. It potentially misses some large rare effect alleles, but the question is, are rare effect alleles that important for climate adaptation traits? Because if it's an adaptation that's spread in dry environments in the last 10,000 years, maybe it's not actually going to be that rare. Because we're looking at things that actually have adapted in a relatively short time, and so we would expect the frequencies not to be that rare. But we have some discussion of whether that's important. And sometimes, I'm not going to go into the details, you can find the wrong allele with environmental GWAS due to negative linkage disequilibrium with ascertainment bias issues. We can also ask, have we adequately sampled maize genetic diversity? The answer is probably not. We've calculated, our partners in this at UC Davis have calculated, if we pick 500, because maize is a huge population size, there's been loads of recombination. In theory, we would get 95% of all variants if we did whole genome sequencing of 500 well-chosen accessions. We only have 200, we're missing some of the variation. Again, do these minor variants matter? Probably for climate adaptation, not, but if you're looking for disease resistance, maybe yes. And so we're working on obtaining the 500 whole genome sequence lines, and then the PHG can be developed that has most of the variation in it and be extremely powerful. And we're looking into this common versus rare issue. What I want to focus on for most of the rest of the talk is deployment. So what are we trying to achieve with the deployment here? Our starting point, we have some lines that are predicted to have the best adaptation for particular climate variables. Some of these might have QTL markers at all, or they might have just been found in other ways. And we have lines where we've concentrated a high concentration of GWAS hits for a particular environmental stress. These two groups of lines may or they should overlap. Also, with the concentration of GWAS hits, we can cross these lines we've selected with a high number of positive alleles to try and enrich them further, and we should do that. The traditional approach of using this data would be to validate these candidate GWAS hits and then use marker-assisted selection to integress them. But what we found here is hundreds of small effect markers, and we've got hundreds of lines containing them, and we want to do this quickly. So the question we're asking in this project, and the focus of the rest of this, is can we enrich and validate at the same time as we proceed through the integration process? Can we make this faster? Instead of spending time validating hundreds of QTLs doing marker-assisted selection, can we enrich, validate, and integress all at the same time? And the answer, probably many of you are thinking, is probably not. That seems a bit ambitious, but I'm going to give it a go. Okay. Oh, I have longer than I thought. That was I was actually rushing through the slides, and I realized I've got 10 minutes. Okay. So this is the challenge. Can we move multiple variants for a quantitative trait at the same time into elite material, validating as we go? So just first of all, would a brute force approach work? So basically, we make an enormous population, try to introgress everything at once. So what we're trying to do for all QTLs is remove the undesired donor regions, minimize the flanking donors, so cleaning up the linkage drag around the target QTL. And we know that no backcross is going to contain all the target QTL. It's going to be a distribution. And also, more importantly, many of the backcrosses will contain target QTL, but also undesired donor regions from maybe another target. So, and then we still have to validate the QTLs. So we'd have to have an unfeasibly large population to do this, spend a huge amount of money. Can we do it faster? And so what we did was, so we want to go fast. We want to validate and enrich as we carry out integration. And we are going to accept, if we start with 200 QTLs, we're going to lose most of them in the process. This is a quantitative approach. Just throw in 200, see if we can get 10 or 20 that come out at the end. It's still likely to be more rapid than if we validated and integrate them all separately. But can we do this? And we had our funders, the Gates Foundation, arranged for a discussion on this, bringing experts from around the world on this. And we had a good discussion of this, although there was lots of disagreement about the strategy, I think it's fair to say. And so we tried to come up with approaches we might think would work for this and discuss this with partners. And various people who were at that meeting are here and had really good advice on what we can do. So what do we want to do to make this work? Well, we need to enrich. We need to clean up the backgrounds. We're going to have to genotype frequently because if we just keep on crossing, we're going to lose QTL. We want to assess the QTL status at the same time as validating. So we can validate if the markers are still segregating, but also we need to see if they are still segregating in the first place. So I'm going to spend some time on this diagram. It's not going to be easy to explain. This is one strategy, and it fits into a pool of strategies that we're trying and working on simulations. So on your left, we have the main breeding program, 2 heterotic pools, test crossing, 1, 2, elite yield trials, hybrid yield trials, and so forth. So I'm going to focus only on one pool at a time. So let's say we're starting out with 100 gene bank donors carrying lots of markers that we're interested in. So for pool 2, so we're going to look at 50 of them for pool 2 here, and we're going to cross those to 10 elite parents. So each of these elite parents is crossed with 5 of our donors, and these donors contain a range of alleles, but probably, the way we've selected them, any one positive allele is likely to be in 2 or 3 donors because these are stacked sets of enriched donors for alleles of interest for a particular stress trait. So then we will make some F1s. We'll make 200 F1s, cross them again, recurrently cross with the 10 elite parents to get some. And we may go to BC2 or BC3. And our breeders have already started doing this in maize. So then 200 times the 50, BC2 or BC3. When we get there, then we do the F1. We start doing some testing. And what we're thinking here is, so what we will have here, these introgression steps have been made in stress environments. So we're going to lose some of the QTL. But what we want to do at this stage is evaluate individual lines. So if a line happens to, which one of these lines is better for yield and what QTLs do they contain? At the same time, we look crosswise and say, well, we have 1,000 lines and we have this allele for this particular marker that we know is there is in 100 and the bad alleles in 900, the positive alleles in 100 and 900 lines don't have it. Is there an effect that we can see here? And some of those effects will already have happened in the early part, because if they weren't effective here in the stress environment, they're gone, or they had some huge linkage drag issue. So this is a test of whether the QTL is effective, and also whether we've created some good material. But we need to, then we select from the best of these, and then we want to stack further. See, I was 5 minutes ahead. Now, this bit is a bit complicated, but I haven't shown it in detail because one of our collaborators on this project is going to publish this, and we didn't want to put the details because it's kind of but this is a rapid cycle augmentation. So what this cycling does here, and this is used for marker-assisted selection as well, it's a pre-breeding augmentation cycle with alternating steps of calculating GBVs to clean up the background, mixed with selecting for the alleles you're interested in. And it's crossing multiple alleles into a cycle so that you have maybe 7 to 10 alleles of interest that you're trying to select on being augmented in frequency in a population, combined with other alleles, and having the background cleaned at the same time. This cycle is key to this process, but I don't have time, or I don't really want to until it's published, go into the details, but I could talk about that if people are interested. But basically, it's augmentation, cleaning, all at the same time. And at the end, you get a line that's fixed for several useful QTLs, and you get several lines that are enriched with different combinations of QTLs, and you're going to test them here in this bridging material. You're going to test this in the general breeding program test cross trials to see if these actually have the resistance you think they have, the resilience to the stress that you think they have. There's a lot of questions on this. I probably don't have much time to go into this. So these are just a summary of the selection of questions we have to ask. How many elite parents are bridging? Which elite lines? So are we going to use for the bridging? Do we want to cross? We probably want to cross into a high value yield elite, but one that's not super drought resistant to begin with. Otherwise, we can't see the complementation of the drought resilience. Which and how many donor by elite combinations? How many backcross and selfing? Just the logistics of it. And also, how are we going to confirm this? So obviously, we want to look at the G by E, so we have to do the test crosses in optimum and stress environments. But what do we want to compare against? Because we need a baseline here. So maybe we have to have some lines that were the elite, but not introgressed as the comparison. We want null introgrescence. What is the introgression strategy, which you've seen before, but we can vary that. What is the way to maintain and increase so even if we get to this point, then we have to have a strategy for maintaining the allele frequencies in the elite population, and this is a problem that we have across CG for general marker-assisted selection, and Eduardo and others have been working on extensively. And how do we include this as a routine process? And basically, we're starting the simulation work now on this, because this is a lot of heavy-duty simulation work. To wrap up on deployment, so for crops where the elite population is significantly differentiated from the landrace gene pool, like maize, and for highly polygenic traits, most of the novel variation we find is now going to be large effect genes. How do we use that variation is the question. We can continue doing what we've been doing before, use lines that are predicted to do well from landrace field testing, environmental GWAS, etc. This will continue to be useful, but this is a slightly slow process. Can we do it faster? Well, we have QTL available from QTL mapping of the stress-adapted lines in environmental GWAS. We know some of those are false positives, but we don't care because we don't want to do all 200. We just want to get 20. And so we want to find a strategy to do this as rapidly as possible. We're going to lose much of that along the way, but we think this will work, but we still need simulations to demonstrate this. And a key point is that we need to very carefully consider the design, evaluation, and validation steps along the way, because we also need to check if it's working. So we put forward a strategy here, but we also have to put all the steps in place to make sure we're actually achieving what we want to achieve. So this is ambitious, but this is, I think, where we have to go now. That a lot of extract from the gene banks. And I'm going to stop there.